I'll start at 5 after. All right, I think we can go ahead and get started. Good afternoon, everyone. Thank you for joining us and welcome. It's a great pleasure to introduce today's seminar speaker, Professor Philip Iversen, who earned a Ph.D. in statistics from Iowa State University. After a year in Australia as a visiting faculty member at the University of Newcastle, he went to work for Eli Lilly and Company. Most of his 25-year career at Lilly was spent in preclinical oncology research. He's one of the original authors of the Internal Asset Guidance Manual at Lilly and is an associate editor of the Asset Guidance Manual ebook published by the NIH. This manual of more than 1,400 printed pages focuses on assays for high-throughput screening and lead discovery. It provides practical information, including guidelines and best practices that are not available anywhere else, and serves as the go-to resource for drug discovery scientists in public and private sectors throughout the world. Among many contributions to this community, Phil and his colleagues developed several important statistics which have been widely adopted. The signal window, reported in 1997, provided the first metric to adequately assess the quality and reproducibility of an assay that was intended for high-throughput screening. The Z-prime factor statistic reported in 1999 is based on the same concept, an assay signal window adjusted for assay variability. The minimum significant ratio, or MSR, reported in 2006, enabled the comparison of potencies to guide chemists and biologists during lead optimization. The MSR also identified assays that lacked the reproducibility to provide such information. While both of these tools were developed within the pharmaceutical industry, Phil and his colleagues disseminated this information as the NIH began its 10-year molecular libraries program, which made small molecule discovery tools available to the public sector. Since then, the information and statistical tools that Phil and his colleagues contributed have had a major positive impact on the quality of drug discovery science done throughout the world. Phil is currently Associate Professor of Statistics at Luther College in Decorah, Iowa. His presentation today is titled Reproducibility and Replicability, Definitions and Methods. So please welcome Phil.
Thank you, Nathan. I am attempting to share my screen and start my presentation. You'll have to let me know if you can see it.
Yep, you can see it.
Excellent. Okay. Well, thank you to Sita and Nathan for the invitation to speak with you today. And thank you, Nathan, for that introduction. I will note that that was much longer than I wrote for my introduction, but that was very well done. Thank you.
Thank you. There's a lot to say about you and your contributions.
It's been a long time. So I want to speak with you today about a very important topic. I think it's important, reproducibility and replicability. And I'll focus on some definitions and methods that I know about. There's certainly other things we could say about this topic, and I'm sure there are things I don't know. So I welcome conversation, hopefully some time for Q&A at the end, but if a question comes up in the middle, feel free to ask and we can stop and talk about it. Okay, so I think the title slide has all been covered, so we'll go on. If I can advance. There we go. Okay, so what is the importance of reproducibility, replicability, et cetera? I think none of us need to be convinced. There have been a number of high-profile publications in recent years and not so recent years. Just a very short list here. I suspect most of you have seen at least some of these. And I will talk about some of the contents a bit as we go, along with some other things besides that list. And so my high-level outline is to first cover some definitions, both from the ICH, the International Conference on Harmonization, and more recently the NAS, the National Academy of Sciences. They published a consensus report on reproducibility and replicability in 2019 that I've just recently come across, thanks to Sita and Nathan. But I have some highlights and some summaries of that report and some comments to make about that. And then I will share some some examples of statistical methods that Nathan mentioned that are in the assay guidance manual and that can be applied to measuring reproducibility and replicability and the other words that start with RE and end in ability, like repeatability, etc. Okay, so here we go. Some first definitions: accuracy and precision. I'll bet most of you are familiar with this, and some of you have even seen a image like the one on the right that distinguish between these two important but different notions of accuracy and precision. So the idea of the picture is that maybe you're shooting arrows at a target and you're trying to hit the middle. And so accuracy is how close the measurements are to the true value, how close you are to the middle of the target. That is also known as bias, which is kind of the opposite of accuracy, or one minus the accuracy, if you will, but it's the same concept. For example, how close is your sample mean to the true unknown mean in an estimation setting? Precision, on the other hand, is how close the measurements are to each other, regardless of how close they are to the truth, regardless of how close they are to the middle of the target. So the upper right target, for example, has high precision, but low accuracy. Usually, in the data we've collected in our scientific experiments, we can only measure precision. We don't know the truth. We don't know the true value of the thing we're trying to measure or to estimate. Well, and we must know the truth to, formally measure accuracy. There's at least one exception to that: calibration assays, where we have known standards, and we can measure both precision and accuracy in that sort of context. Some review of an overview of some ICH guidelines for analytical procedures. To the best of my knowledge, the most recent guidance on that is from 1996, and there's a link there. And that document describes some different types of precision using some of these words we're talking about today. So repeatability in this document is defined as within run repeatability, within run precision. So it's the precision under the same operating conditions and in the same lab over a short interval of time. Sometimes that's also called intra-assay precision. The next level up is intermediate precision. So that's more about between run variability, but still within the same laboratory. But how consistent are your results on different days, possibly with different analysts or operators within the same laboratory, different equipment, such as light readers, et cetera, dispensing equipment. That's intermediate precision, according to the ICH. And by the way, we'll see that these definitions are not universal across all labs and fields of study, but this is one set of definitions. Reproducibility, then, is the between lab precision. And we could talk about agreement between laboratories. That is how, well, precision and agreement are kind of the same thing, really, but near synonyms, you might say. But that's about between laboratory agreement or precision. Robustness is another related topic to all of this that is covered in this ICH guideline. And a definition there is the capacity for an assay system to remain unaffected by small but deliberate variations in the method parameters, whether that's your incubation time or your exact incubation temperature or amount of reagents. You can do multi-factor designs to measure robustness. I'll show an example of that later, where you intentionally tweak some of the conditions of your assay over a narrow range to see how consistent the results are. And that can provide an indication of reliability during normal usage of the assay, where you can't guarantee that things will be exactly the same from one day to the next. Okay, here is the National Academy of Sciences report from 2019 with a cover page and a link. So reproducibility and replicability in science. Pretty big topic. I'll just highlight a few things from this report that I found interesting. First of all, there's no consistent set of definitions of these concepts across all fields of science. The NAS report references this Barba paper in 2018 with 3 different sets of definitions of reproducibility and replicability. Version A is no distinction between the terms, but notice in the table on the right, This paper found that such usage, that type of usage in political science and economics. The set of definitions in B1, reproducibility refers to computer codes used to regenerate the results, is what reproducibility is about, whereas replicability refers more to experimental replicability or reproducibility, where a researcher is collecting new data to try to reproduce those same or similar findings. And this paper found the use of those definitions across a wider range of fields of study, as you can see in the list on the right, and closer to the kinds of things you're involved in, I think. But there's the B2 category, which is a little bit different. Reproducibility refers to independent researchers arriving at the same results. So that's more like between laboratory reproducibility, or other researchers using their own data and methods even. So that maybe even speaks to a kind of robustness, perhaps. Not even trying to do it exactly the same way, but can you get a similar result? That seems like a higher bar to me. While replicability refers to a different team arriving at the same results using the author's original artifacts. It's an interesting choice of words, I guess. I haven't read the actual paper, just quoting what is in the NAS report. But that seems like trying to do it the same way as the original team, but still a between laboratory kind of thing. And there are two fields that paper identified that uses those definitions. And I would guess this isn't even universal, right? The B1 definitions might not be universal within biomedical research, for example. So we have these concepts, maybe nebulous concepts of being able to reproduce results, and we have different terms and different definitions for them. But they're all part of the same set of ideas and goals to be able to get the same results or similar results later on that you got before, in order to advance science and scientific knowledge. Continuing with some highlights of the NAS report. So given that there are those different definitions out there, this report is trying to, and is proposing a set of definitions for moving forward. So here is their proposed definition of reproducibility: obtaining consistent results using the same input data, computational steps, methods, and code, and conditions of analysis. In some ways synonymous with this idea of computational reproducibility, and Sita added for me instrumental reproducibility, maybe another important and related topic. You know, when I first saw this, and I've come across this in other recent years, especially in the field of statistics and statisticians talking about reproducibility of analysis, this is what they're talking about. And I'll say more about that on another slide, but I just want to acknowledge that this might be a new idea to you, this focus on reproducing the analysis itself with the same data. I'll say more about that in a moment. But moving on to the definition of replicability in the NAS report, obtaining inconsistent results across studies aimed at answering the same scientific question, each of which has obtained its own data. Okay, I think this is maybe more about the ideas you might have in mind under the concept of reproducing your results and having a consistent knowledge base for advancing scientific knowledge. So here's a quote as part of this definition: "Two studies may be considered to have replicated if they obtain consistent results, given the level of uncertainty inherent in the system under study." I think there's some nebulous aspects to that statement that I'm also going to say more about in a moment. It sounds good on the surface, but what do we really mean by replicated? And what do we really mean by consistent results given the level of uncertainty? I'll give you a couple examples that occur to me when I think about what that might mean. There's also this notion of generalizability, which they are saying is the extent that results of a study apply in other contexts or other populations that differ from the original one. This might sound like robustness, but I think it's distinct from that. And I'll come back to that possible difference later and my thoughts on that.
Phil?
Yes,
May I ask a quick question?
please.
So these three concepts are big in terms of data sharing, which is going on currently, how much people are beginning to share data from their publications. There's a big Nature article, I think, just came recently about sharing data, because there's a lot of issues about people not sharing data, raw data, as well as processed data in publication, so that people cannot reproduce, using the same analysis, or people cannot replicate that information, uh, so that they can actually go back and redo the same kind of analysis or same kind of experiments and reproduce and replicate. So I think this is an extremely important concept here. But I do like the question I have for you is, um, in your work with analyzing all the clinical data, you get clinical data and preclinical data from different places. Don't you try to reproduce that information or reanalyze that information and make sure that the conclusions are correct? Isn't that part of the things that we used to do when we had, and in fact, we are doing that in NCATS when we get independent data from other labs. We are trying to figure out whether that data is reproducible and replicable. So my question to you is, isn't that a common practice in the pharma industry?
It is. It's certainly becoming one, definitely in recent years, and maybe going back 10 or more years or so. First, a correction to what you said.
Yeah.
I haven't looked at much clinical data since my first few years at Lilly.
Okay.
Clinical, but you may have meant preclinical, or you may have
Preclinical.
meant, and those lines are blurring because there are clinical samples with genetic information being generated from them that are now being analyzed in preclinical studies.
That is
For the
correct.
purpose of
Yes.
finding biomarkers, right? You all know about that.
Yeah.
So yeah, the lines are blurring. What do you mean
So,
by clinical data?
so Sid and Phil, we always politely call that trust but verify.
Yes, 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 or trust and verify. Yes, right. But to your point, more to your question, we're getting bigger and bigger data sets, starting 10, 15 years ago, and reported analysis results are, it's not as clear perhaps how someone got from their data to their analysis results. And it is increasingly important to be able to reproduce the analysis given the data,
That's
because
right.
the data has gotten bigger and more complex, and the
That's right.
analysis methods have gotten more complex.
That is
And
correct.
how do you know when you're reading a biomarker paper that the authors did an appropriate analysis?
Yeah, I think the recent Nature comment talks about people not only, even though journals are requiring that the authors provide raw data to somebody who asks for it, or they need to deposit, most of the people are not providing that information.
Well, yeah, and there's lots of
Even
conversations to be had there
yeah,
about what to do. Should
yeah,
that be a requirement?
yeah. What is the
Yeah,
incentive to do that? Anyway, that's a separate
yeah,
topic, but I'll
yep,
let you
yes.
go
Oh,
ahead.
lots of directions we could go with this.
Yeah.
That's right. That's right. So I do like this move towards assigning some word like reproducibility to this notion of computational and analysis reproducibility. That is an increasingly important topic for the reasons we just stated. Okay, very good. So my next slide, so here's my take, the next few slides on these NAS definitions. So starting with reproducibility, so we may not, this may not be how many of us use that term, the computational part, but I think it is important. It is consistent with these recent notions of reproducible research that we've maybe read about, and computational reproducibility, et cetera. As I said, more analysis is now done via computer programs and code, and sometimes very long and complicated analysis code, especially in genomics, as an example. Analysis details and decisions about the analysis, including what data to exclude or how to impute missing values, et cetera. Lots of decisions can get buried in the code, and transparency is important. So another whole other topic is, how well is your code commented so that someone else can follow what you were doing? So I'll conclude this slide by saying having that code available that can reproduce the analysis results is better than just using point and click software. And the bigger the data set, the more important that is.
Yep.
Yep. Okay. My take on the replicability aspect, consistent results across studies. The NAS report acknowledges that is hard to define. Here are just two examples that occur to me to help us think about this at maybe a basic level. Example A, suppose you did a study, number one, where you observed an average of 60% tumor volume reduction versus a control, and the p-value is 0.01. And you do another study with that same design, same treatment groups, and you observe 51% tumor volume reduction versus control, and the p-value is 0.06. Have you replicated that first result? On the one hand, I would argue yes, because the average percent tumor volume reduction is pretty similar in the two studies. Some of us might argue no, because one study is significant at the 0.05 level and one isn't, but they're awfully close. Mm-hmm. And I would argue that there's not much difference really between a p-value of 0.01 and 0.06, except for where we tend to put that line in the sand, that 0.05 line. I'm going to say more about that in a moment too.
Phil?
Comment?
What about the effect size?
Well, that's what I'm talking about. 60%, 51%, those are the two observed effect sizes. They're pretty close in my view, in my experience. Those are not very distinguishable. I would call those consistent.
Mm-hmm.
Yep.
Yeah, okay, yeah.
Yep. In contrast to that, suppose your study one in my example B, suppose study one was the same result, but in study two, you observed 85% tumor volume reduction with a P of 0.005. Well, now you might argue, yes, I've replicated that study because I got a significant result both times. We could also say that they're both effect sizes are over, say, a minimum relevant threshold of 50%, for example. On the other hand, 60 and 85% are farther apart in actual effect size.
That's what I was getting at, yeah.
I think that's exactly what I'm getting at. That's my point.
Okay.
How do you decide?
Yeah.
Right. If there's a big enough difference, it's more clear. If it's a really small difference, then it's also more clear that you have replicated. But where's the middle ground? There's gray area. So these are things to think about with replicability. But here's what's more important to me than focusing on individual studies, and the report says this too. It's not about individual studies. It's about the bigger picture. And we might say, to borrow a legal phrase, preponderance of evidence. That's more important than any individual study. And let's dig into that idea for a few minutes. So speaking of not focusing too much on one study, here's a quote. Personally, the person writing this quote, prefers to set a low standard of significance at the 5% point. A scientific fact should be regarded as experimentally established only if a properly designed experiment rarely fails to give this level of significance. So the writer of this quote is taking that bigger view, saying most of the studies you've done need to be significant to establish a scientific fact. So who said this? Ronald A. Fisher in 1926. I think it's safe to say that Fisher invented the p-value. And this is where we get our 0.05 cutoff from. Here's a paraphrase of this quote from a statistician, a statistics professor at Texas A&M from 2015. A paraphrase, one instance of p less than 0.05 was never intended to be the final word in any investigation. Okay, speaking of p less than 0.05, here's another quote from Ronald Fisher from 1956. No scientific worker has a fixed level of significance at which, from year to year and in all circumstances, he or she rejects hypotheses, or they rather gives their mind to each particular case in light of their evidence and ideas. So Fisher updated his thinking 30 years later and said it doesn't always have to be 0.05 for your cutoff for significance. All right, moving on. Back to the NIS report and my take on the definition of generalizability. To me, that's about consistent results across assay systems and or across assay conditions. One example, test tube to mouse, mouse to human. To me, that's an example of generalizability. Or another example, can you obtain similar results for significant biomarkers or important, let me say important biomarkers that you find via different assay methods, but looking for the same kind of biomarker in the same disease state, for example. So to me, that's what I think they're talking about in terms of generalizability. And so that's not the same as robustness. Robustness is usually focused on a single assay system and how reproducible is that system under small but deliberate changes in the method parameters. Here I remind you of that ICH definition of robustness. So robustness, I think, is usually focused on a single assay system. Generalizability is speaking to consistency of results across assay systems. Okay, I want to spend some time now talking about some specific statistical methods targeted towards these concepts, things that we developed at Lilly and are now published in the assay guidance manual and in some separate papers that Nathan referenced in the introduction. So I'll start with this idea of a replicate experiment study. And what we call it isn't that important, but more important what it does. So this is a kind of experiment primarily for in vitro assays to measure their replicability and upfront validation, statistical validation of a study. And this is similar to the notion of ICH repeatability. We can also use it for between lab comparisons. And so the notion of generalizability, this kind of study can be used to start to get at that notion as well. By running the same assay in two different labs or similar assays in two different labs that are trying to measure the same thing. So the standard guideline is more or less 20 compounds that have a range of activity. Or if your assay capacity per run is lower than that, oh, sorry for the typo there, then we'll do that. Larry Strand, head of the core assay group at Lilly way back in the day, coined the phrase flexible standardization. So we have some standards, but we need to be flexible to enable ourselves to implement them on different assay systems. So if you can only do 6 compounds in a run, then do 2 runs of 6 with those same 6 compounds. But they should be independent runs in the sense that you've done them on different days, ideally, and with a completely different setup. You go through all the steps of running the assay, preparing the reagents and the compounds, et cetera. Well, that does raise the notion of, do we want to be able to separate the biological variability in the assay from the variability of the compound preparations? That's another topic I wasn't going to discuss today, but it can be an important one. Hopefully your compound prep reproducibility is low, but we did do a study on that once at Lilly where we found some issues, and there were some easy corrections in terms of weighing protocols and so forth. Okay, but I digress. So the analysis is what's called a Bland-Altman analysis. It's paired data. We have something like an IC50 or a potency value or an efficacy value for each compound run twice in 2 different runs. So we have that paired data. The usual Bland-Altman analysis makes a plot of the differences of those paired data points plotted on the vertical axis versus the mean of each pair of numbers on the horizontal axis. But this is an example using potency data where a log scale is more appropriate for doing means and differences. And that's equivalent to plotting the ratio on the vertical axis with a log scale versus the geometric mean on the horizontal axis, again with a log scale. So each data point in the plot is one of those 20 compounds, or in this case, there were 25. And what we're looking for is kind of a uniform scatter around the center line at a ratio of 1. A mean ratio of 1 is the ideal. And that the mean ratio in this data set is very close to 1 at a value of 0.97. And then given that, that's the first thing. And then the second thing is we want as little scatter above and below that line as possible, and sort of as uniform a scatter as possible, with no patterns or no evidence of higher variation at one end of the potency scale versus the other. And that's pretty well satisfied here. Your visual acuity systems, your eyeballs might be seeing a slight downward trend there, but I would argue that's not important. It's not an obvious or extreme concern. The next thing on this graph I'll point out are the green lines. So those are the so-called ratio limits. That's simply a 95% confidence interval for the mean ratio, And so that's fairly narrow because means have lower variability than their individual values that go into them. And then the red lines are showing the limits of agreement, and those are calculated as the mean plus or minus 2 standard deviations of the individual data. And so that's measuring the spread across the individual compounds. And we have a criterion for that, that the limits of agreement should be between 3 and 1/3. So no more than 3-fold variation in a 2 standard deviation margin in each direction. And so this assay, this particular data set passes that criterion for the limits of agreement to be between 1/3 and 3. And then we also calculate an MSR from these data. So MSR stands for minimum significant ratio. That is the smallest ratio between potencies of 2 different compounds that we can say is statistically significant at the 0.05 level. That's how we define that. And so that's a number that characterizes the reproducibility of the assay in terms of the potency value, that key performance indicator, that key outcome that we care about for being able to distinguish between the activity of different compounds within an SAR, for example. So with an MSR of 2.77, then if 2 different compounds differ in their potency by 3-fold, then we know that's a statistically significant difference. Now, there's an important point here that the MSR calculated from this 2-run experiment is actually a within-run MSR. It's only measuring within-run variability. So I won't go into the statistical justification for that, but that's an important point to make. You really need more than 2 runs to measure between-run variability. We'll show an example of that in a moment. The p-value here is for that paired t-test for the mean ratio being different from 1. And so it's good that that's a large p-value. The mean ratio is close to 1. So there's a consistency on average between the 2 runs for this set of compounds. And because we have these paired data, we can also make a scatter plot. We'd like to see a nice correlation there. The magenta line is the y equals x line, the line of agreement. Not a least squares fit, but the line of agreement. So we can judge how close these pairs of IC50s are to each other for each compound between the 2 runs. So if you want to read more about this, here's a link to that. I think it's that 2006 paper that Nathan mentioned at the beginning. Here's another example where I actually took the same data, but introduced a shift between the 2 runs. So you see in the scatter plot, all the points are now above the line of agreement. So this is to illustrate what can happen when there's a problem of agreement between the 2 runs. So now the mean ratio is 4.8-fold on average between the runs. So that's the blue line at about 4.8. We've shifted away from the ideal mean ratio of 1. We still have the same amount of spread, so the MSR stays the same. But the limits of agreement have shifted, and now the upper limit of agreement is much bigger than 3, and that's a warning flag for the reproducibility tests. And now there's a very significant difference in the mean ratio away from the null hypothesis of 1, we could say, and the preferred ideal value. So that's the replicate experiment. A couple of examples, replicate experiment study. The next method I'll talk about is control charting, and that gets at the idea of intermediate precision, the precision across runs within a lab. And to generate this kind of data, you just need 1 or 2 reference compounds that are tested in every run or every few runs, depending on your capacity. But then we need more than 2 runs. Many more, what does many mean? Well, at least 6, but more like 10 or 20 to start to get a good idea of the intermediate precision or between run reproducibility. So thinking about a potency assay, in vitro potency assay, we can plot the log potency versus run number, run ID, or run date. And we can learn a lot by looking, just by looking, watching for patterns and shifts, et cetera. And now we can estimate what we call an overall MSR, an MSR that captures both between run and within run variability. Gives us a more realistic picture of the performance of the assay. So here's some example data from a long time ago at Lilly and anonymized. We don't know what compound or assay this is. But here are some IC50s plotted versus time for one compound, the reference compound. You see some variation from run to run, but the mean seems fairly steady, you might say. And the numbers are ranging from about 80, probably micromolar or millimolar, up to, or a micromolar or nanomolar maybe, up to 300 or 400 perhaps. And that kind of variation translates into a moving MSR that looks like this. So it's hovering around 3, an MSR of 3 on average. That's that green reference line is simply located at 3 for a reference point. So what is a moving MSR? Once we have 6 runs, the first 6 runs, we can calculate the standard deviation of those log IC50s, and that feeds into the MSR formula, and we get a number. So this first point in the graph on the right is the MSR from the first 6 IC50s. The second MSR in the graph on the right comes from the 2nd through the 7th IC50s, and so on. So we have this moving window of 6 consecutive runs from which we calculate an MSR, and that gives us an idea of the shifts in the assay reproducibility over time, and also the long run stability of the assay reproducibility as measured by the MSR.
Bill?
Yes.
Can I ask a quick question?
Please.
So this MSR concept, I think, can be used to compare 2 independent assay methods that are measuring the IC50 of a set of compounds. Is that correct?
Yes. Yeah, if you have 2 different assays that you're
Yes.
comparing and you're wanting to ask which one is more reproducible, and the primary outcome is a potency,
Yeah.
you can do this for efficacy too, but then it's a minimum significant difference.
Yeah, minimum 6. Yeah, I
But yes, to your question, comparing 2 assays, absolutely, you can.
Yeah.
But
Because
then I think the unanswered question is, how big a difference in MSR is statistically significant?
Yep. Yep.
And a separate question is, how big a difference in MSR between 2 assays is practically meaningful?
That's right. That's right.
I don't know the answer to those questions, although we could work on that. We could work on that. But if you have one assay whose MSR is 3 and another one whose MSR is 5, let's say, then I think you have a pretty clear choice. You'd prefer the assay with the smaller MSR.
Yeah, exactly. That's
3 and 3.5, I'm not sure there's that big a difference.
Yeah. No, sometimes
it might come down to ease of use and cost and et cetera, and capacity, et cetera.
Sometimes we are forced to change the assay methodology due to technology or you know, different labs or something like that. So that's what I was asking that question. So it's very useful in that sense as well. Yeah.
Make a change to a given assay. You want to assess the impact of that change.
Or you change the reagent, antibodies change or
exactly
something like that.
Right.
And then you can
what happens to your reproducibility
exactly
6 runs later.
Yeah.
Yes.
My understanding is that at Lilly, this moving MSR concept actually allowed the assay biologists to understand things that were happening that you weren't necessarily apparent from the MSR values themselves, right?
Yeah.
Yes. Even just this run chart of the reference compound potencies goes a long way to helping you detect issues as they're beginning to occur.
Yeah, we have detected in the old days people changing, the MSR will change, running the same, the instrumentation changed, some reagent changed.
Yeah,
So many
right,
things that happened we were able to detect with the moving MSR.
right. I should point out there's an important caveat with moving statistic charts, whatever your statistic is that you're charting. But a moving window chart does suffer from the following issue. If you have a single unusual point, like this IC50 here on the left, higher than all the others, not extremely, higher, but stands out a little bit. That will tend to cause cycles in the moving MSR chart, because there are, as you can see here, it might be this one, or I'm not sure. Yeah, probably this high section here of this moving MSR chart. Those 4 or 5 MSRs probably include that one higher point in their window. And because of this moving window, you can get these artifacts that look like cycles that really are not, we shouldn't overreact to. They're not that important to worry about, but they can be caused by a single, somewhat extreme point like this one. So just be
Thank
aware
you for
of that.
pointing that out.
Yeah.
I can totally see what you mean now, but I never noticed that before.
Yep. That was a big deal when I first learned that, I don't know, 12 years ago or something. I was not aware of that for a while, but it makes sense once you see it, once you have it explained. You see some numerical summaries down here for the IC50 summary chart on the left. There's the highest value, the lowest value. Overall just means the average of all of them. And then current is the latest number. And then those same 4 numbers for the moving MSR chart. So it's kind of nice to see a 3.6 as the overall MSR for this full set of data on this assay. When we look back, we made a database of all the MSRs for all our assays at Lilly, most of them, all the ones in the centralized screening lab at least. And after many years, we were able to look back at that. We found that the median MSR across all our assays was somewhere around 3. So when you look at the assay guidance manual and our publication on MSR, we say that the criterion for a good assay is 3. That's a line in the sand, like the p-value, that we should take with a grain of salt, right? There's this notion in this field of reproducibility about suitable for the intended purpose. Is your assay suitable for the intended purpose? And sometimes an assay with an MSR of 5 is suitable for the intended purpose because you may be screening and looking for bigger differences to advance your SAR. That's just another example, I would say, of flexible standardization. Take the standards, but tailor them to your, and understand, think carefully how to apply them to your situation in a way they make sense.
Your colleague Devin will often speak about the MSR at assay guidance workshops, and one of the points he makes is that if you have a higher MSR, it just means that you need to repeat the experiment a few more times.
Yes, that's a thing. Let me talk about that. Yes, so if you have an assay whose MSR is 5, and the standard way of calculating that applies to the scenario where you're testing each compound once, right? And which is what you typically do when you're screening, or even maybe an SAR. Some compounds you might repeat because they're interesting and you want to confirm what you got, look for consistency for that compound, that's fine. But if you have an assay with a higher MSR and you want you want to lower the MSR for all compounds, then if you have the capacity and can afford to do it, test every compound twice in different runs, and then analyze the averages. The average of the 2 runs for each compound. And there's a formula. We can tell you exactly what the MSR is for averages. But off the top of my head, if the MSR for single run per compound is 5, then the MSR for the average of 2 compounds, if you're testing all of them twice in separate runs, then it drops to somewhere around 3.4, I think. There's a pretty big bang for your buck in going from 1 to 2 runs per compound. So that's what Nathan was talking about, Devin was talking about. Yes. That's a useful thing. All right, what's our time? I think we're looking good. Oh, no, we're not. Keep going. Okay. Database MSR. So if if you're running an assay frequently and you've got many runs, and either by happenstance or by design, preferably by design, you have multiple compounds tested in 2+ runs, even if it's 10% of them, you can use a repeated measures analysis of variance to calculate both within run and overall MSR. And that's the most robust, if you will, or most reliable measure of MSR for the assay, because it's using more compounds and more runs than either of the 2 previous methods. Won't go into the details on that, but we've developed ways to do that. Here are a couple graphs of that kind of data. So each row of points is a different compound. They're plotted in an order such that they're in the order of maximum IC50. And so it makes this kind of line, squiggly line here. But then you can see the spread of each compound back to the left. And you can identify the reference compound down here in the left corner because it has a lot more runs than the other compounds. This assay is maybe showing some higher variability for the less potent compounds. But this assay looks more consistent spread across the range, although maybe less spread in the lower quarter, a little more spread in the upper 3/4. But and here the overall MSRs are shown below. So 5.9 on the left, 3.4 on the right. So the assay on the right is a tighter assay. But let me keep going here. The multi-factor factorial design I talked about as a method for robustness. I think in the interest of time, I'll skip over the details of that, but there is a paper here. Sita sent me this paper many years ago, and it's a good example of intentionally tweaking the conditions to check for reproducibility and robustness. And they got greater than 90% yield over all the experimental runs because they were varying things only a little bit. So there were 3 factors and 4 different responses, but the focus being on yield. But everything's plotted over there on the right. So let me skip over that so I can talk about this last topic, which I think is more important. This slide is just summarizing those methods I just described to you. And you'll have access to the slides in the recording later. So let me just spend a few minutes. Oh, I'm sorry, 2 more topics. Briefly, comments about in vivo assays. It's a really good idea to include a positive control in every run. And if that takes up too much capacity, do it every few runs so that you can begin to apply some of these methods that I've just described for in vitro assays in an in vivo setting. It's also a good idea to occasionally repeat other active treatment groups. Maybe you have an open slot next week, so repeat something you did a couple weeks ago. That gives you at least some data about reproducibility in your in vivo assay system. And if you do that, then you can apply those control charting and database analysis methods I just described for assay reproducibility. Okay, then on another topic of looking at the bigger picture, this is a figure from that Economist article, How Science Goes Wrong. And I wanted to mention this for a few minutes here because it's kind of an oversimplification. It's just, these calculations are all based on a very basic hypothesis testing scenario, statistical hypothesis testing, where you have a 5% false positive rate. That is your p-value cutoff for significance is 0.05, and 80% power, which is the same as a 20% false negative rate. And so over in the diagram here, suppose you have 1,000 different scientific hypotheses that you're testing, and suppose 100 of them are true. We don't know which 100 of them are true, but suppose 100 of them are. And you can redo this with 200 true hypotheses out of 1,000, or 50, or 500. The calculations are the same, the results are different. And I'll show you a couple of different examples. But the idea then is that among your true hypotheses, 20% of those will be false negatives. You will miss your data, your experiment will misidentify, or you'll miss finding the effect 20% of the time because you've designed your studies to have 80% power. You can improve that if you do 90% power, but here's the illustration. So the reds are the false negatives, the true hypotheses that you miss discovering. The dark green are the false positives. Those are untrue or false scientific hypotheses that you end up claiming are true because you got a p-value less than 0.05. You get 45 out of 900 false positives because you have 900 false hypotheses, and 5% of those will be false positives. Okay, so we can summarize all that in this so-called confusion matrix. 100 true hypotheses, 900 false ones. And then 80% of the true ones will test positive. That means your experiment, your data will show a significant result. Test negative means not significant. Here are the false positives down here, 45 out of 900. But a different calculation is something called positive predictive value. So let me be clear about this. False positive is the probability of a negative test. False positive is the probability of a positive test given the hypothesis is false, 45 out of 900. But positive predictive value is the reverse question. What's the probability that your hypothesis is true given that you got a significant result in one experiment? That's a different calculation. And in this case, it's 80 over 125 or only 0.64. So what this means for reproducibility is that if you get a positive result in one test and you try to reproduce it under these assumptions, you only have a 64% probability of reproducing that first study. Now, this is not exact. This is a qualitative calculation, but still important to remind us to not overreact to any single negative study that didn't reproduce an earlier result. Okay, here's another example. What if you keep everything the same, but reduce your false positive rate to 1%? What if you lower your p-value cutoff for significance to 0.01? Well, then the positive predictive value goes up from 0.64 to 0.899. And in fact, in recent years, the American Statistical Association has published an article, I think this is 2015, recommending that we lower our default cutoff for significance from 0.05 down to 0.01 or even 0.005 as a new default. Now, will you miss more things? Yes, but this positive predictive value is an important calculation in the mix. Here is another way of looking at that, and this is from Appendix D of the NAS report, and it describes what they did in great detail. It's very similar to that diagram in the Economist article, kind of looking at the same kind of question. It's using Bayesian inference calculations, which that are giving a little bit different set of numbers, but it's the same qualitative story, the qualitative conclusions. It's still a hypothesis testing context. Given a p-value and a prior plausibility of the alternative hypothesis, that's like that prevalence or percentage of true hypotheses among every hypothesis you're testing. That's what prior plausibility is. Then you calculate the posterior probability of the alternative. That's the Bayesian version of that positive predictive value. It's the same concept, even if it's a slightly different calculation in this appendix versus the Economist article. And so the key takeaways are, if you have a new discovery, hypothesis 1, let's say, that's relatively surprising to you, that corresponds to a lower prior probability of that hypothesis being true. And in that case, then it is actually less likely to replicate in your next experiment. It has a lower posterior probability of being true given your next set of observed data. Actually, that's given the current observed data. But both of these examples speak to the notion that we shouldn't be too concerned about a single study not reproducing a result. We need to look across multiple studies, preponderance of evidence. But as with the Economist example that I modified for you on the previous slide, this graph, you can take away that lowering the p-value cutoff, that is going from 0.05 to something lower, moves you up for a given prior probability of a hypothesis, moves you up the scale for the chance of reproducing it. I think that's really important and really interesting. And if you don't understand all the math behind it, we can talk more maybe at a later date. But let me just wrap up here with some final thoughts. Depending on how much reproducibility work you're doing now or not, you might be wondering about the cost of doing this work, of assessing replicability, reproducibility, et cetera. My answer to that would be, we can't afford not to do it. And that's what the article about the economics of reproducibility on my first slide in that list of papers talks about. Another way to think about that, if we don't have time to do it right now, when will we have time to do it over? But what is reasonable for how much of your total resources should you spend on this work? I argue that doing nothing is not an option. 50% is clearly way too much. The optimal might be around 5 to 15%. I'm not sure. It's probably in that ballpark, more or less, depending on your circumstances. Are you in the final stages of an SAR where you want to be really sure what you're moving into the clinic? Or are you doing exploratory early work where it's less critical? Be flexible. So finally, ramping up replicability work over time. I'll quote a colleague of mine from Lilly, Steve Ruberg, when he said, think big, start small, and move fast. So I will stop there and thank you for your time.
Phil, thank you. That was fantastic. I'd like to open the floor for questions. Questions or comments? Phil, I can begin. One question I have, you were talking earlier about algorithms used to analyze data, right? And, and seeing, you know, huge increases in the use of machine learning algorithms.
Right.
And you pointed out how critical it is to understand, you know, the basis for decisions being made. And I think in some of those cases, you know, for those algorithms, it's not possible to know exactly how the algorithm's working.
Well, I would hope that it could be, even if it isn't now, if we were more intentional or required people doing this work to share their code, first of all, but even better, share well-documented code. And maybe also in there, in an appendix or a supplement to a published article, include more details, written details about their analysis algorithm and the choices they made. Steve Ruberg did a presentation back at Lilly when I was still there about, he had an example of a published work finding a biomarker, but when they dug into it, they found out that they removed some data for this reason and removed some other data for that reason, and they got down to, like, I don't know, 12% of the original data. No surprise, the result didn't reproduce.
Yeah. No, I think that there's a big issue with reproducibility and replicability and all of that. There are a lot of people using machine learning and AI to do a lot of predictions about what it is, but the quality of the data that they are putting in is in real question.
Garbage in, garbage out.
Yeah, exactly. So this is where I think somebody said all models are wrong. Some are good.
Some are George Box, professor of stats at Wisconsin. Yes.
So the thing is, that's what I'm worried about, that people are just barreling into AI and ML right now using existing data, which is really
a lot of people are. And I was encouraged when I saw an article recently that whose topic was more data is not always better.
Well, what do they mean by more data? That's the question.
Well, that's all relative.
Yeah.
What is big data? Is it hundreds of thousands of rows? Is it millions of rows? Depends on who you ask.
Yeah.
Or how many columns? Do you have 35,000 columns because you've measured every gene. That's also big data. And 20 rows. 20 people,
I'll send you the Nature article about data sharing. It is really eye-opening because nobody's sharing data. I mean, even though,
is that true, or just lots of people are not sharing data, and some are?
There are lots of people sharing data, but important information is missing.
Right.
So then nobody's sharing data. That's my point.
Not well enough, right?
Yeah, that's right.
Yeah.
They say, some people say, yeah, they will share data, then they won't. Some people say they share data, they share the data, it's lots of things are missing. So it's a big article. I'll send it to you guys in case you're interested in.
All right.
Journals are demanding to share data, but everybody's saying they are, but it's at various levels there.
You know, maybe we need some standards for how to share data, protocols or something.
There's not only standards, but there needs to be some incentive. Somebody needs to fund it. Then they will.
Right.
And it's a more cultural issue than anything else, but that's okay.
You've heard about the Reproducibility Project?
No, which one is that?
I have to search it here.
There's a journal that started coming in out of Oxford, and then they suddenly disappeared, called the Reproducibility Journal.
Yeah, I'm not sure if I'm going to find it, how quickly I'm going to find it here, but I can email you later.
Okay, that's fine.
It started from a researcher at a cancer, I think an academic, maybe cancer center in Florida, who challenged a esteemed author's results and got fired for it, and has been ever since raising these concerns about reproducibility. It's been a while since I've heard about it, so I can't remember exactly what it was called. I'll see if I can find it.
Okay.
Wow. Yeah, thanks.
Yeah, she spoke at Lilly, I think, is where
Oh.
I first saw that, or else at a conference somewhere.
Anyway,
Anyway,
thank you.
I'll let you know.
I gotta go for another one now.
Yeah, me too.
Talk to you later.
Yeah.
Okay, bye.
Thanks,
Thank
Phil.
you. Excellent
Thanks.
talk.
Take care.
Thank you.
Thanks,
You're welcome.
Chrissy. Thanks, Sita.
Thank you. Bye.
Bye.
Bye.